Hello students, let us learn about cell migration and cancer metastasis in this lecture. Let us first see the introduction. Tumors that occur frequently in older animals and humans are of small size, usually localized and delineated by a fibrous capsule. Such tumors are called benign tumors and are of little risk until and unless they interfere with normal functions of the body or secrete excess amounts of biologically active substances like hormones. In contrast to such benign tumors, tumor cells may become malignant, also called cancers, and are characterized by active growth and division, as well as invade the surrounding tissues, enter the body's circulatory system, and proliferate in areas away from their site of origin. This spread of tumor cells and establishment of secondary areas of growth is called metastasis, and most malignant cells eventually become metastatic, although they may be localized at the initial stages. Sometimes the malignant cells can be distinguished from the normal cells by microscopic examination, as the malignant cells usually have rapid growth rate, high nucleus to cytoplasm ratio, prominent nucleoli, many mitoses, and with relatively little specialized structure. Cell invasion and metastasis are two major characteristics that differentiate the malignant or metastatic tumors from the benign ones, and they also form the two important life-threatening aspects of cancer responsible for the mortality and morbidity of cancer. During tumor metastasis, the tumor cells that acquire the ability to penetrate the surrounding tissues initiate invasion of the basement membrane and extracellular matrix, followed by penetration of the lymphatic or vascular circulation, also known as intravasation. The metastatic cells move through the circulatory system and invade the vascular basement membrane and extracellular matrix at a new location to proliferate secondary tumors by the process of extravasation. The malignant tumors derived from endoderm or ectoderm are called carcinomas, and those derived from mesoderm are called sarcomas. Most tumors are solid masses except the leukemias, which is a class of sarcomas that grow as individual cells in the blood. Let us now see the cellular properties of metastatic tumor cells. The first property is cell-to-cell adhesion. The cells of the primary tumor mass in epithelium-derived cancers are held together by intercellular structures and cell-to-cell adhesion, and mutation or dysregulation in these structures may lead to the dissociation of the primary tumor cells and invasion and spread of metastatic cancer cells to secondary sites. The key cell-to-cell adhesion structures that maintain the coherence of cells are adhesion junctions, including desmosomes, tight junctions, and gap junctions. In epithelial cells, the adherence junctions provide the most powerful form of adhesion, followed by the tight junctions and gap junctions. A key protein involved in maintaining cell-to-cell contact inhibition in normal epithelium is E-cadherin or cadherin 1 or CDH1, which is a member of the cadherin family of proteins. In cancer cells, the cell-to-cell contact inhibition is lost due to the loss of E-cadherin and promotes uncontrolled cell growth and metastatic dissemination of cancer cells. Desmosomes form adherent points in cell-to-cell junctions by linking integral membrane proteins such as desmocollin and desmoglein to intermediate filaments via desmoplackins such as plakophilin and plakoglobin. Desmosomes also play a role in tumor suppression, and downregulation of desmosomes have been observed during tumor development and early invasion. The tight junctions are the gasket-like cells that surround the apical pole of each columnar epithelial cell and play important role in maintaining polarity by separating the apical and the basolateral membrane domains physically, and also prevents interchange of materials between the luminal and antiluminal tissue fluid compartments by diffusion along the paracellular pathway. Claudin family of proteins forms an integral component of the tight junction and may play a role in human carcinoma development. The expression profiles of claudin have been reported to change during tumorigenesis, and upregulation or expression of certain claudins in aberrant tissues may affect the metastatic potential of cells by directly altering the structure and function of tight junctions, and may also affect cell signaling pathways. Another cell-to-cell adhesion protein that could serve as an oncogenic molecule is beta-catenin, and beta-catenin signaling may target claudins. Gap junctions are unique cell-to-cell channels made up of connexins in vertebrates through which diffusion of small metabolites, secondary messengers, ions and other molecules takes place between neighboring cells. Connexins appear to play a role in cancer cell dissemination during the late stages of tumor progression. The next is cell matrix adhesion. Adhesion between cells and the interaction of cells with the surrounding extracellular matrix, also known as cell matrix adhesion, involves the binding of cell surface integrins with the protein components of the extracellular matrix. The functional integrin proteins consist of a heterodimer structure made up of different alpha and beta subunits that have different affinities for different matrix proteins. Many integrins have been linked to metastatic likelihood, and some cancer cells may deposit certain extracellular matrix proteins that enhance metastatic progression. The selectins, for example, P-selectin, E-selectin, and L-selectin, are adhesion molecules that bind circulating leukocytes to vascular endothelium during inflammation in response to injury or infection. Selectins have been observed to regulate adhesion of circulating cancer cells to the walls of blood vessels. Integrins and selectins play a role in progression of various types of cancer, including colon and lung carcinomas and melanomas. Selectin may mediate arrest of tumor cells and adhesion, contributing to metastasis, while integrins may mediate interaction between the tumor cells and the surrounding environment, which contribute to cancer progression. Focal adhesion kinase, or FAK, paxillin, and CD44 are some other key adhesion molecules that are potential targets for antimetastatic studies. Next is cell migration. Cellular migration is a trait essential for cancer progression and metastasis, in addition to its role in normal development and homeostasis. The increased migration of cells is an initial key step in the metastatic cascade that leads to the scattering of cells and migration from the primary tumor site to secondary tumor sites by penetrating the basement membrane and extracellular matrix through intravasation and extravasation of vessels. Several proteins that inhibit cell migration and proteins or pathways involved in enhancing cellular motility play essential role in cancer progression, and they represent a potential strategy for controlling the spread of metastatic tumor cells. There are two main patterns of cancer cell invasion by migration of tumor cells crossing the barriers of the extracellular matrix to spread into the surrounding tissues. The first one is the collective cell migration, and the second one is the individual cell migration. Next is collective migration. Collective migration involves the migration of group of cells interconnected by adhesion molecules and other communication junctions. Such collective cell migration has been observed in the development and progression of many cancers. In collective migration, the cancer cells of the tumor mass detach from the mass and penetrate into the surrounding tissues in multicellular groups, forming invasion structures. During invasion, the protruding tumor cells can take the actin cytoskeleton using integrins to form focal contacts, where proteolytic degradation of the extracellular matrix takes place, leading to the creation of a space for invasion of the tumor tissue. Next is single cell invasion. Single cell invasion involves individual migration of cells that occur via two different types of movement, that is, mesenchymal and amoeboid type of movements. Cancer cell undergoing mesenchymal mechanisms of cell invasion change their morphological properties and acquire mesenchymal cells phenotype and invade the surrounding tissues after detaching from the tumor mass. The malignant cells which use the mesenchymal type of movement lose epithelial polarity and become elongated and spindle shaped like the fibroblasts, and therefore this type of invasion is called fibroblast-like migration. This type of migration involves many cellular molecules and involves cycles of 5 specific sequential steps. The first one is the formation of a protrusion called a lamellipodia or a philopodia on one of the cell poles by contracting actin cytoskeleton with the help of under small GTPase RAC1 and CDC42 and beta-1 integrin proteins. The second step is the formation of focal adhesion at the contact site between the cell and the extracellular matrix with the help of beta-1 and beta-3 integrins. The third step is the assembly of focal contacts at the cell matrix interface involving integrin-mediated interactions and activation of proteolytic enzymes or matrix metalloproteinases, serine and threonine proteases, cathepsin, leading to the destruction and remodeling of the surrounding extracellular matrix. The fourth step involves change in the actin cytoskeleton polarization mediated by myosin II and resulting in cell body contractions. The fifth is the pulling of the trailing edge through the remodeled matrix. Next is amoeboid mechanism of cell migration. Amoeboid mechanism of cell migration is similar to the movement of single-celled amoeboid organisms, and it is the most efficient mode of migration of single tumor cells. When single tumor cells spread to the surrounding tissues using molecules and destroy the extracellular matrix, they turn to the amoeboid mechanism of invasion. During amoeboid migration, the tumor cells attain round or elliptical shape with fast deformability and adaptability and penetrate through the narrow spaces in the surrounding extracellular matrix through successive high-speed cycles of expansion and contraction of the cell's body, involving the development of blade-like protrusions of the cell membrane. Generation of changes in the cell shape involve cortical actin cytoskeleton and are controlled by small GTPases RHOA and its effector ROCK kinase. The mesenchymal and amoeboid movements have some degree of plasticity and may shift from one migration type to the other, that is amoeboid to mesenchyma and mesenchymal to amoeboid transitions under the influence of certain changes in the activity of certain cell molecules and the need to adapt to tissue microenvironment conditions. The next is the epithelial mesenchymal transitions. Epithelial mesenchymal transition is the process by which epithelial cells change to mesenchymal phenotype by undergoing a series of morphological and biochemical changes. Although epithelial mesenchymal transition is a normal feature during development, it is also involved in the establishment of invasive and motile cancer cells phenotype required for metastasis. The process of epithelial mesenchymal transition generally involves the loss of cell-to-cell adhesion due to loss of epithelial cell junctional proteins such as E-cadherin, claudins and ZO1, which leads to the formation of a polarized epithelial morphology. Next is the tumor microenvironment. The progression of malignancy in cancer does not involve only the change in genetic material of tumor cells, but also involve many molecular networks of interaction between the non-malignant connective tissue cells, for example, cancer-associated fibroblasts and tumor-infiltrating immune inflammatory cells, and cancer cells within the tumor microenvironment. Activated cancer-associated fibroblasts secrete various growth factors, chemokines, collagens, and enzymes that modify extracellular matrix in both paracrine and autocrine manner, and these molecules supply an altered three-dimensional scaffold of extracellular matrix and communication network that supports proliferation of cancer cells, leading to tumor invasion and metastasis. The tumor-infiltrating immune cells, such as the tumor-associated macrophages, myeloid-derived suppressor cells, dendritic cells, tumor-infiltrating T cells, regulatory T cells, and mast cells are recruited by chemokines and cytokines secreted by tumor cells. In addition, tumor cells also activate mast cells, promotes the expansion of the myeloid-derived suppressor cells, and the polarization of tumor-associated macrophages, and also inhibit DC maturation through IL-10 secretion. All this lead to changes in the immune cells of the tumor microenvironment, leading to tumor proliferation, survival, invasion, metastasis, angiogenesis, and the remodeling of the extracellular matrix. Now coming to the conclusion. Metastasis and cell invasion are the two most important features that distinguish malignant tumors from benign tumors. Cancer cell invasion involves progressive infiltration, invasion, destruction, and penetration of the surrounding tissue. The term metastasis is used to describe the ability of tumor cells to establish new tumors at a secondary site away from its primary site of origin. The important events of metastasis include detachment of the tumor cells by downregulation of E-cadherin, attachment of tumor cells to matrix components mediated by receptors, degradation of extracellular matrix, migration of tumor cells to the circulatory system, and establishment at distant sites by extravasation of tumor cells involving adhesion to endothelium followed by egration through the basement membrane mediated by proteolytic enzymes and adhesion molecules. Thank you.